Hi everybody, so I'm here to talk about IAs in biology, and I have provided to Miss Bailey, and you guys probably have in front of you right now, an example of an IA with some teacher comments on it. And this is something that the IB provided. It's got the title, I'm going to read off my screen here, A Study on the Effect of Smoke Water on the Germination and Growth of Eucalyptus pilularis. You may have never heard of that type of eucalyptus before, but what's great about this lab is that it just lays out in a really logical way how you could set up a practical IA. So just to highlight a few things, if you're looking at the page in front of you or the screen in front of you with this lab on it, there are sections obviously like research question and background and those sorts of things that are very standard to Group 4 IAs. But there's also a preliminary setup where in this lab, as I just go through it, they have a test that they want to do. And in order to set that up, they have to run a preliminary experiment. And so what that does nicely is that it shows that they had to go through not just one experiment, but actually two experiments, so it's multi-layered, which means that their personal engagement is strong. So that's one good way to get a full 2 marks in your personal engagement. They also have laid out everything quite nicely in terms of what they needed for the experiments and how they were run. One warning, as you're looking at this example, this is for plants. And sometimes for our IAs, if we want to use plants in biology, you might actually have to start doing your research a little bit before our 3-day moratorium at the beginning of January in a few months. And so just keep that in mind, because plants sometimes need time to grow depending on what it is you're trying to do. For the rest of the paper that you have there, please take a look at the observations, always quantitative and qualitative, and also take a look at how they've processed the data. Obviously in biology, you know our stats. We've got chi-squared, we've got t-tests, Spearman ranks, we've got the ANOVA, which we will talk more about in class, and all those other beautiful things like standard deviation, mean, and all the rest of it. Those are some things that in bio, it's good to have at least one of them. If you don't, it's okay, you can still make it work. But please think about those type of things for your processing, along with any graphs or percent differences or anything like that you might want to test. When you get down, you can see this particular report when it gets down to the conclusion, laying it out really nicely and focusing even equal weight on your strengths and weaknesses can help a lot in terms of giving your conclusion and evaluation a really full and thorough approach to assessing your lab and how it went. I hope those few points add to anything I've said before, and keep in mind that online on ManageBack in our labs section under the file tab, there's a whole bunch of support material there for writing IAs as well. Thank you very much.